The aim of this procedure is to demonstrate the successful establishment of human fungiform taste papillae cell culture. This is accomplished by first obtaining the fungiform taste papillae from human volunteers and dissociating them using an enzymatic cocktail. The second step is to transfer the cells from the human fungiform taste papillae into a cell culture dish. Next, allow the cells to grow and change the media to maintain the cultured human fungiform taste papillae cells. Ultimately, calcium imaging, confocal microscopy, and light microscopy are used to demonstrate the presence of taste cell markers and physiological responses.
The main advantage of this technique is that it allows the use of cell lines instead of freshly collected tissue each time one undertakes an experiment.
Though this method can provide insight to human taste mechanisms, it can also be applied to other systems, such as other species and other studies of regeneration.
This method can help to answer key questions in the taste field, such as how taste cells regenerate and what factors regulate gene expression during taste cell proliferation and maturation.
To begin this procedure, obtain the human fungiform taste papillae by removing it from the dorsal surface of the anterior portion of the tongue using a pair of curved spring microscissors. The details of this protocol were published in a previous Jove article. Then, place the sample immediately in the isolation solution. To digest the fungiform papillae, isolate them in the isolation solution with collagenase type 1, elastase, and soybean trypsin inhibitor in a 35°C water bath with circulation and gentle oxygenation with 95% oxygen, 5% carbon dioxide for 30 minutes. After incubation, wash the papillae with Ringer solution. Triturate it with a fire-polished glass pastel pipette 10 times. Then, centrifuge it for 3 minutes at 2500 rpm at room temperature. After that, remove the isolation solution. Add 1 ml of taste cell culture media to the papillae. Then, transfer the digested fungiform papillae to a glass dish. Now, dissect the fungiform papillae gently with a surgical razor. Add 250 microliters of the dissected papillae onto the cover slip coated with rat tail collagen type 1 in the cloning cylinder. Afterward, add 1 ml of taste cell culture medium to the well. In this procedure, incubate the plate at 36°C in a humidified incubator containing 5% carbon dioxide for 2 days. Then, change the complete medium. Taste cells may not be visible in the first 3 days, but they will eventually bind to the coated cover slip. Next, remove the cloning cylinder from the plate and the medium completely. Add 1 ml of taste cell medium to the well. After that, replace 1/3 of the medium every 6 to 7 days. In the meantime, check the cells under the microscope every other day. Most of the new cells grow underneath the cell clusters. Cell clusters usually detach from the surface of the cover slip after 2 to 3 weeks in culture, leaving the newly generated cells. Once 40 to 50% of the cloning cylinder is covered by the expanding taste cells, trypsinize the cells using 0.25 weight per volume trypsin EDTA for 2 to 3 minutes at 36°C. Then, transfer the cells from the well into a 15 ml tube. Subsequently, dilute the taste cell culture medium 4-fold with the medium containing fetal bovine serum. Centrifuge it at 3000 rotations per minute for 5 minutes at room temperature. After that, remove the supernatant and resuspend the cells with 1 ml of taste cell medium. Now, transfer the cells to a T25 plate. Then, add 4 ml of taste cell medium. Maintain the cells at 36°C in a humidified incubator containing 5% carbon dioxide. Next, replace 1/3 of the medium every 6 to 7 days until the cultured taste cells have reached 100% confluence. To enable taste cells for harvesting, wash the cells once with sterile PBS. Then, trypsinize them using 0.25% weight per volume trypsin EDTA for 2 to 3 minutes at 36 degrees Celsius. Repeat the centrifugation as described previously. Then, resuspend them in taste cell complete medium. Afterward, transfer the cells to a fresh T75 flask and replace 1/3 of the medium every 6 to 7 days. When the cells are close to 100% confluence, split the cells to no more than 1 to 4 dilution in a T75 flask for maintaining adequate growth of the cells over time. To freeze the stock of primary taste cells after trypsinization, add 3 ml of complete taste cell medium. Transfer the cells to a sterile 15 ml conical centrifuge tube. Then, centrifuge at 2500 rpm for 5 minutes at room temperature. Carefully remove the supernatant and gently resuspend the cells with an appropriate volume of freezing medium containing 95% fetal bovine serum and 5% DMSO. Next, transfer the cells to a labelled sterile cryovial. Cap the vial tightly. Then, place it in a freezing container containing isopropanol and place the container in the -80 degrees Celsius freezer for at least 1 day before transferring to the liquid nitrogen. To thaw a vial of frozen primary human fungiform taste papillae cells, place it in the 37 degrees Celsius water bath in a laminar flow cell culture hood until just thawed. After that, transfer the cells and freezing medium to a sterile 15 ml conical centrifuge tube. Add 5 ml of taste cell culture medium to it. Next, centrifuge the cells at 2500 rpm for 5 minutes at room temperature. Carefully discard the supernatant and gently resuspend the cells with complete taste cell culture medium. Then, transfer it to a sterile T25 tissue culture flask. Cells within these cultures display many molecular and physiological features characteristic of mature taste cells. Gusteucin and phospholipase C beta-2 mRNA were detected by reverse transcriptase polymerase chain reaction, and the products were confirmed by sequencing. The expression of gusteucin and phospholipase C beta-2 was detected immunocytochemically, indicating the presence of type 2-like cells. Cultured cells also exhibited robust increases in intracellular calcium in response to appropriate concentrations of several taste stimuli. In these studies, sweet and bitter stimuli elicit an increase in intracellular calcium that may correspond to a depolarization.
Once mastered, this technique can be done in 1 to 2 hours if it is performed properly.
Following this procedure, other methods, such as calcium imaging and immunocytochemistry, can be performed in order to answer additional questions like what taste receptors are present or what is the rate of proliferation in response to certain growth factors.
After watching this video, you should have a good understanding of how to dissociate and grow human taste cells.